So in this video, I'm going to be talking about glycolysis. Glycolysis is the first step in cellular respiration. So what happens in glycolysis? So the big picture is that glucose goes in, 2 molecules of pyruvate, 2 ATP, and 2 NADHs come out. The reaction occurs in the cytosol, and it's regulated by allosteric inhibition of phosphofructokinase by ATP. Okay, so we're going to talk about all of this. Glycolysis is a sequence of 10 reactions, and it occurs in the cytosol. The first 5 reactions are called the energy investment phase. And what happens in these reactions is we actually spend 2 ATP to begin to start to make more ATP. The next reactions, the next 5 reactions, are called the energy payoff phase. And so in these reactions, NADH is made. NADH is an electron carrier, and ATP is produced in a process called substrate level phosphorylation. What is substrate level phosphorylation? It's when you have a substrate that has a phosphate group, and that phosphate comes off and goes on to ADP to make ATP. And the net yield of glycolysis is 2 NADHs, 2 ATPs, and 2 pyruvate. So I'm not going to expect you to memorize the 10 steps of glycolysis and all the intermediates. And what I want to start to do as we kind of march our way through what happens during the oxidation of glucose is I want you to be able to follow the carbons and see how those carbons become different molecules over time. Okay, so we're going to represent all of our carbons with these little black circles, and we're going to follow those carbons throughout these processes of glycolysis, pyruvate processing, and the citric acid cycle. Okay, so here's our 6-carbon glucose. And in the first step of glycolysis, glucose is phosphorylated by enzyme 1 to make glucose 6-phosphate. Okay, so this took 1 ATP to make this molecule. We then have 2 more reactions, so this molecule of glucose 6-phosphate isomerizes, so changes shape, and it becomes fructose 6-phosphate. And then the next reaction, we spend another ATP to phosphorylate this again to make fructose 6-bisphosphate. Okay, so now we spend another ATP. So this is the energy investment phase of glycolysis. We've spent 2 molecules of ATP. Fructose 6-bisphosphate is then split in half, and these 2 molecules, this DHAP and this glyceraldehyde 3-phosphate, can isomerize back and forth into each other. But eventually, this G3P will continue down the pathway of glycolysis and become pyruvate. It gets oxidized into pyruvate in a series of reactions. And because we have 3 carbons here, which will then turn into 3 more carbons here, this will happen twice per glucose molecule. So that's how we get to our 6 carbons. So during these last reactions, substrate level phosphorylation occurs to generate NADH and 2 molecules of ATP. But recall I told you this is going to happen twice per glucose molecule. And so we're actually going to make 2 NADHs and 2 ATPs per glucose molecule, or 4 ATPs per glucose molecule. But keep in mind that we already spent 2 glucose molecules up at the top here of this pathway, and so our net is 2 NADHs and 2 ATPs. Okay, so just to walk through again, we spent an ATP, we made 4 ATP, which leaves us with 2 ATP. And this pathway is regulated by feedback inhibition, okay, at this enzyme 3, okay. So here's enzyme 3, it's called phosphofructokinase, and it has an active site that accommodates ATP and fructose 6-bisphosphate. When that gets into the active site, they react, and then they break apart. This enzyme also has an allosteric site for ATP, and when a lot of ATP builds up in the cell, it binds to this allosteric site and shuts off this enzyme, so this pathway shuts off. So when there's a lot of ATP, we don't want to spend the energy to make more ATP, so we shut that enzyme off. So we're losing this reaction where we add that extra phosphate. So to review what goes in glucose and 2-ATP. What comes out? 2-pyruvate, 2 NADHs, and a net of 2-ATP. ATP is made by substrate level phosphorylation, and the NADH will go on to the electron transport chain. We'll talk about this in a couple days. This reaction occurs in the cytosol, and feedback inhibition by allosteric binding of ATP by phosphofructokinase shuts off the pathway. And I'll end with a little pitch for why I care about glycolysis, and that's because the cells I study primarily rely on glycolysis for their energy. That means that they don't make a lot of ATP because that's only 2 net and 2 NADHs. They don't make a lot of energy. They don't make a lot of protein. But what they can do is that they can function in the absence of oxygen. So if you are fighting an infection and there's not a lot of oxygen in your tissues, these cells can still do their thing. And that's all I have to say for this video. And thanks, bye.